Biotechnology. Welcome to Cases Study Hub. In this video, we will be learning the differences between genomic library and the cDNA library. So, the first difference between the genomic and the cDNA library is that in genomic library, we consider all the possible fragments of the DNA, whereas in cDNA library, only expressed gene sequences are taken. Now, because that only all the possible fragments are taken, the data will be, or the genome like we have is, which is large and complex. Whereas because only express sequences are taken, the data will be small as well as manageable, right? And it represents the entire genome of an organisms. Only est refers to expressed sequence tags, the sequence which will be like expressed during that है ना, only coding sequence it will be represented. Now in this, the expression of a particular gene from the genomic library is very difficult because there is no splicing की ਹੁੰਦਾ है, like the proper whole DNA we have, it has two parts, coding as well as the non-coding sequences. When these non-coding sequences are removed, the process is known as splicing. ठीक है, only coding sequences are directly expressed in the prokaryotic system because splicing occurs in the eukaryotes. So next is vectors used because we have a large amount of the data. So we need plasmids, cosmids, phages, BAC and DIAC. These are the BAC and DIAC are basically bacterial artificial chromosome, yeast artificial chromosomes. Lambda phage, plasmid, cosmid, these are can be used because we have to accommodate a large fragments. And for the smaller fragments, you can use the plasmids, phagemids, and the lambda phage. Phagemids the change hai, and here cosmid and BAC. Okay, plasmids and lambda phage can be used for both. The enzyme used for genomic one is restriction enzyme, and here it is reverse transcriptase, and is used for the whole genome, or like gene mapping will be in whole genome. So gene mapping and evolutionary studies, what changes are being taken place from the previous ones, right? And here it is used for the gene expression and protein production because only express sequences are there and they will code for the protein and the functional genome we studied in this. So this was the whole topic, genomic library and cDNA library.